we can see slightly raised structure in the reptile, but we can see more development and larger volume of this space, and more capacity has been developed in the humans. And we can see that the skull portion, okay, where the brain sits, to which we call as a brain case, that portion is huge, and that is what is contributing to the complexity that we can unravel or undertake as an individual. Okay, so its origin lies here. So the later development, lot of things have changed, but early development, most of the things are similarly. So we can find that these parts are found. So these are the arches that develop. So one of these arches is what we call as the pharyngeal arches, and how the pharyngeal arches develop in these different organisms were also studied by Heinrich Rathke. So comparative analysis can give you this kinds of distinct developmental aspects that are happening in the organisms. Okay, so similarly we can find other areas how they have developed. Okay, so when we look at the early development and we look and compare lizard and humans, they look more or less similar, isn't it? So you can see the head structure and the vertebrate structure more or less is in the same fashion, and you can see the somites that are lying. You can see here also the somites that are lying, and you can see the tail like small tail like structure that is there, isn't it? So these all, you can see the embryo accumulating from the limited egg yolk that is there, the nourishment, and here you can see the egg yolk that is, it is connected. So amniotes, isn't it? So how amniotes accumulate is through the connection through the placental cord, isn't it? So cheek, especially cheek and humans have this amniotic kind of a linkage, and you can see placenta that is there. So these kinds of commonality you can find in early embryonic development, gradually which changes later on. So this is the important observations that has confirmed us the developmental principles, uh, and which has resulted through the process of epigenesis, gradual building up process. Okay, so by this observation, this epigenesis principles have got reiterated and reinforced, and that is why we have accepted epigenesis as a theory. Now, what August Weismann did was, visually, graphically, we are trying to display what his kind of principles was stated by him actually. So Weismann's nuclear determinant kind of a concept, or germ plasm kind of a concept. So what he's saying is that the cytoplasmic plasm where the nucleus lies is what we call as the germ plasm, where the nucleus, the main content element lies. So it is having distinct elements. So it has been shown graphically as colored star, red star, green circle, triangle. So imagine this to be the genetic elements. And what has been mentioned by him is that during cleavage they get segregated, and as the cells divide, each of these elements get picked up, and there is a mosaic like of arrangement, and based on what element it has picked up, it would characterize and get specialized. So we see that the green element it has picked up has resulted in this differentiation of this particular structure on this cell, is what is the diagrammatic way in which to understand what August Weismann observed. And what we have mentioned is the tunicate observation, that in tunicate the macho factor, the yellow pigment, uh, contributes, has been accumulated by the cell and contributes to muscle later on in the development. So that is has reinforced his principle. And again, what has been shown by uh, Wilhelm Rocks, who was a prodigy or son, the student of Weizmann. He supported Weizmann's theory that there is a mosaic model, and what he did was a experiment on frog embryo. He just took a two cell stage frog embryo after post fertilization. This is the zygote, and this is two cell stage. He poked a needle, red hot needle, at one of the two cells, and the cells were formed by longitudinal cleavage and which were lying side by side. So suppose this is left and the right, he has poked in the left hand side, and what has happened is the left hand side cell got damaged. So later on, what was the normal cell on the right hand side have undergone cleavage, and multiple cells accumulated on the left hand side, and what has resulted is in the only half embryo which was resulting from the right side surviving cell, which was not harmed. So an embryo developed from that half. So if there is a mosaic kind of a model that is running, whatever the principles or elements or germ pools that were necessary to form the half embryo was has survived in the right hand side cell in this two cell stage, and that has contributed to the half embryo. And whatever half embryo was to be contributed from the left hand side was lost because of the damage to that left cell in this two cell stage, and that's why the other half embryo couldn't be developed. And this kind of an observation reinforced the mosaic model or kind of a development. And mosaic kind of model tells that the elements get segregated, the germ pool gets segregated, whichever germ pool is present in the zygote gets segregated in each cell, and then that's why they get this different characteristics, and each cell has a different characteristics. So this is not true for all the cells and all the development, and this was proved wrong. And Hans Reich was that person who has proved it wrong, and he did an experiment that we call as the recombination experiment. Recombination not in the sense of the genetic recombination, but recombining cells. Okay, so recombining cells of the early embryo. So there is cleavage, and there is multiple cells that are there. So each cell lies at a particular position. So he just took cells from one position and placed it in a different location. So what we call as the juxtapositioning or jumbling of the cells, and what is the result of it? So we know that if a particular structure has to develop at a particular point, that cell has to lie at that point, isn't it? So when we looked in the previous image, the right hand side developed into a right half embryo. So if that cell, right hand side cell has been picked up and placed on the left hand side, you will get a inversion of the structure, isn't it? So that should be the case if the mosaic model is working, isn't it? So when Hans Dries did such recombination experiment, he didn't get a completely juxtaposed or inverted kind of embryos. What he got was a normal embryo. What he did was just what we call as removed few cells very early in the development and just looked at the embryo. He got a smaller embryo, but more or less the embryo was complete. And even if the position of the early cells to a different location was done, the embryos developed normally. So this indicated that these cells are very plastic in nature, plastic in the sense they are flexible in nature, and they have the ability to transform themselves from one role to another. And what is that other role has not been lost. If even if it is present in the earlier location, if you pick up that earlier location, suppose this cell is there, and if you place it over here, so if it was going to form the right hand side, if you place it at the left hand side, it is